Can you hear me?
Yeah.
Okay, thank you. So let me start my talk. Hi, everyone. My name is Hideki Yamaguchi. I'm a graduate student at the University of Tokyo. And first of all, I'd like to express my gratitude to the session organizers, especially to Chris for his kind of communication via email. And I will talk about Evotuning protocols for transformer-based variant effect prediction on multi-domain proteins. This slide shows a typical variant effect prediction pipeline in supervised setting. Thanks to the recent advances in protein language modeling, such as TAPE, USM, or ProTrans, we can easily obtain general use representations. In protein engineering, we are often interested in a specific target protein. Since experiments such as directed evolution are costly, we are usually given a small number of measured values, typically of order of hundreds or less. This necessitates the unsupervised incorporation of evolutionary information. To this end, we here consider an additional fine-tuning step using homologous sequences to the target protein called Evotuning before supervised variant effect prediction. Let us see the complication posed by multi-domain proteins, which are often of interest in realistic protein engineering. Here we see a protein with 3 domains, the first of which is a mutagenesis target. At the same time, domain A and B are interacting. Thus, their correlation influences the representation of the mutagenesis target, while domain C is functionally independent of these two. Naive mask language modeling for full-length protein can include potentially noisy contributions from the domain C. So our approach is to focus our attention on the most relevant parts, explicitly considering a multi-domain arrangement called domain architecture in a sequence. This is very simple but effective, as shown later. For evaluation, we sourced mutagenesis experiments on 7 proteins classified into 3 archetypal categories: A, single domain proteins; B, proteins with multiple independent domains; and C, proteins with multiple interacting domains. What I want to emphasize is that these proteins have different domain architectures, which needs careful treatment. By combining homology search, using hit sequences in fine-tuning, and embedding strategies, we define Evotuning protocols as in the right figure. For example, in protocol B, we use a mutagenized domain sequence as the query for homology search. As the hit sequences tend to include domains homologous to the target, we use these full-length proteins for fine-tuning. For variant effect prediction, we embed the mutagenized domains. We systematically examined our hypothesis that the proposed protocols A, B, and C are suitable for the protein categories A, B, and C, respectively. The results are as follows. As shown in the table, each Evotuning model outperformed the pre-trained counterparts. Moreover, domain architecture-aware protocols achieved better performances than domain architecture-unaware ones. For example, for YAP65 in protein category B, protocol B, which was considered suitable for the category B proteins, indeed achieved significantly better accuracy than others. If we train the variant effect predictors end-to-end instead of training clusters on top of frozen Evotuned weights, we obtain further performance boost of 5 out of 7 proteins, especially in the data-rich region. Finally, Evotuned models with our protocols worked better than previous methods, such as LSTM-based UniRep or unsupervised MSA-dependent AP sequence. To explore how this substantial performance improvement was realized by Evotuning, we visualized the attention maps of YAP65 models as an example. The map displays a context map-like, roughly symmetric pattern with respect to the diagonal, which is in sharp contrast to the absence of such a pattern for the pre-trained model. This is a bit surprising because no explicit knowledge on tertiary structure was input into the model fine-tuning. This suggests that the structural information was implicitly incorporated into the model, even from a set of unaligned homologous sequences, possibly leading to improved accuracy. To summarize, we defined Evotuning protocols for transformer-based variant effect prediction on multi-domain proteins. The Evotuning models outperformed their pre-trained counterparts, indicating that specializing the general use models to the engineering target is an effective way for achieving better accuracy. We also demonstrated that domain architecture-aware protocols indeed perform the best. The visualization of attention maps suggested transformer models can incorporate tertiary structure information in an unsupervised manner. The complete data and discussions can be found in the article below. If you're interested, please check it. And because I am not able to attend Q&A session in midnight in Tokyo, if you have questions or comments, please send us an email or DM me on Twitter. Thank you very much.